Okay, so we should be streaming live to YouTube now. And well, we're going to start off fine. And so good morning, good afternoon, evening, wherever you are. Welcome to another fun. Okay, so we should be streaming live to YouTube now. And I think I will do that every time I host. So apologize for that. So we're having another fine seminar today, and before we start, I have just a couple of quick announcements. First off, I want to start off by sharing screen and showing you. Noah, can you stop sharing? Thank you. I want to start by sharing a screen and showing you the preliminary schedule for the spring fine seminar. And hopefully everyone can see my screen okay here. As you can see, we have an exciting seminar series in the works. We'll be starting off with March 1st of 2022 and going through the 28th of June in the next series. We have a great number of speakers: Marlene Zuck, Sarah Gendry Parker, Peter Biedermann, Marcus Zottel, Karen Kapheim, Erica van Vanderwall, Louise Evansberger, Trini Dild, Mandy Ridley, Tony Di Ferriero, Gloriana Chavari, Mikael Grieser, Ava Fisher, Karina Logan, Steve Phelps, and Niels Dingemans. And we have a couple of slots that we've got things in the works, and we might have more information for you next week when Carson hosts. We don't have any titles yet, but we just wanted to give you a sense of what's coming up. And I've just learned that another speaker we're having is Lucy Appling will be joining us. Okay, so it's a really exciting series coming up, and we're hoping to have lots more fun discussion. Couple of things. So for those of you who are here for the first time, the seminar runs about 45-50 minutes, and then we follow it up with some discussion. And today we're really happy to have Noa Pinter-Wollman joining us today. Noa is an associate professor at the University of California, Los Angeles. And I think today it's going to be pretty exciting because we're going to hear about her work that focuses a lot on collective outcomes from group interactions. She combines field and lab studies with mathematical modeling or computer simulations and looks at things from the perspective of social networks. She's also been really great at integrating her work into conservation biology. So taking what we learn about animal behavior and learning how that can influence conservation theory, and developing tools for assessing conservation through that lens. Noa has a long history of some really nice work. I forgot to mention she got her BS degree from Tel Aviv University in Israel, master's and PhD from UC Davis. And she's been pretty prolific publishing, doing some really nice publications, over 60 publications in excellent journals, and again, brings another exciting theme to the fine seminar series. In terms of outreach and teaching, she's had a pretty active lab. It looks like a great group, and includes a number of undergraduate PhD students, master's student, and postdocs. And today, Noa is going to be giving us a talk on her work entitled Spatial and Ecological Effects on Collective Behavior. And with that, Noa, you can please take over from here. Thank you for being here.
Thank you so much for the introduction and for the invitation to be here. Let me try to see some of you things. All right. Let's see. And everyone can see my slides. Okay. All right. So today I'll talk about spatial and ecological features and how they influence animal movements and interactions. Oh, where's my computer? Not moving. Okay, there we go. And so we all think about animals and we think about them in their natural habitat. And so we know that spatial features can influence how they move around. For example, they might not always be able to cross rivers, and they might not always be able to cross certain mountain ranges, and they might be attracted to certain places like food places or water holes. But we know that not all individuals are impacted by the environment in the same way. For example, maybe bigger individuals are able to cross rivers while smaller ones cannot. And through this impact on the movement of animals, the spatial features influence different types of animal interactions. So for example, if animals are attracted to water holes, they'll meet certain individuals. If they want to stand in the shade of a tree, they might be forming more bonds with their family groups. And so in different places, they might form different types of interactions and different types of relationships. Now, there's also temporal variation in how spatial features influence movements and interactions. So for example, maybe in dry seasons, you don't meet as many individuals because everyone is spread out because the resources are spread out. And then maybe in the rainy season, animals come together and interact more. So there could be temporal variation in both interactions, but in how the spatial features mediate these interactions. And then there could be feedback between these animal movements and their behavior and the spatial features. So for example, if animals you always use the same areas, you might see formation of animal tracks. For example, here, these elephant tracks that are trails that are formed, and then they can use again, other animals and other species can use as well. So there's this feedback between how spatial features influence where animals move, and then where animals move create spatial features that facilitate further movement, and we get this feedback. And so what I want to have you think about today during my talk is how this combination between spatial features and their influence on animal movements influences social interactions and how they change over time and individual variation in both the influence of spatial features on social interactions and on movement. And when preparing this talk, I was reflecting, I took all these elephant pictures because I have a lot of them from my time as a PhD, and I realized that actually a lot of my PhD fits into these schemes. So I'll go back, a decade or so, almost a couple of decades ago back, and bring a few snippets of examples for how social interactions change over time and have individual variation all interacting with the animal movements and spatial features. And then I'll jump into the collective behavior after some elephant stuff. Here's the feedback. So for my PhD, a long time ago, I studied elephants that were translocated from Shimba Hills, which is down here in the southern tip of Kenya, to Tsavo East, which is arid park, also in the south of Kenya, but further from the ocean. And these elephants were translocated for wildlife management reasons. They were causing human-elephant conflict in Shimba Hills, and they were moved to Tsavo East, where they presumably would not. Sorry, my computer froze for a second. And so what I looked at these elephants after they were translocated to their new home, and I asked how their social interactions changed as they moved from one place, from one type of habitat, which was lush and green, to another place that's quite arid and also has a big local population of elephants. And so this is the first network that I ever published. This is a network of my elephant, the translocated elephants. And what you can see is that in the translocated elephants, these are all, each node is a translocated elephant. The colors indicate animals that were moved together as a family group. And so what you can see is that animals mostly, the elephants mostly interacted with one another, with others that came within their group, but they also interacted across groups. And I wondered whether these interactions across groups, which we know that elephants have because they have this multi-scale society, I wondered if these interactions were more with translocated individuals than with the local population. And I found that indeed the translocated elephant interacted more with translocated elephants than they did with the locals. So this TT is translocated with translocated and TL is translocated with local. And I compared this to a null distribution and what one might expect at random. And these interactions changed over time. So in the first year that I tracked them, the translocated individuals tended to interact a lot more with other translocated individuals, which you see in this bar here. And then in the second year, the number of interactions reduced. Whereas when you look at the interactions with the local population, in the first year, they didn't have many interactions with locals. But in the second year, this changed and they started interacting a lot more with the locals. And basically this kind of immigrant enclave started to kind of assimilate into the local population, which is what we were hoping, what the wildlife management action was hoping for. And of course, there was a lot of individual variation. Not everyone did the same thing. And so when you dump elephants in a place they don't know, some go back. So for example, this bull went from the release site, basically zoomed straight back to Shimba Hill. This happened in less than a week. We had another bull who also was released and then followed this river until the end of the park and then had to go around the people that live along the river and then made it to the coast, but not the coast where he came from, but a different part of the coast of Kenya. And then, of course, some elephants stayed around the release site, mostly it was family groups, but also some bulls stayed in the area. And there was variation in how much they explored this new habitat. So you can imagine that when you come to a new place, you want to explore the area and check what's out there. And how much you explore a new habitat influences how much information you're going to have about the resources there and the other animals that are around you. And so I asked whether this exploratory behavior at first has any relationship with how far they ended up from the release site. And some of the elephants were collared. And so this is data from just the collared individuals. And so it turns out that how much they explored the habitat at first didn't really have any indication for how far they ended up from the release site after a year. So for example, these bulls didn't do much exploration, but ended up very far. And then there were some family groups who explored extensively and ended up really close to the release site. And then some that did extensive exploration and ended up a bit further. But what I did find was that there was a relationship between how much you explore the environment initially and how bold you are. So to determine boldness, I looked at how close the elephants let me approach them with my car. And so the ones that let me approach them closer, so these ones down here, also were more exploratory. So these would be the bold ones that let me approach them pretty closely. And down here, we would have the ones that are more skittish and wouldn't let me approach them much, and they did a lot less exploration. And so one might be able to use this information for future wildlife management actions. If, for example, you're moving individual elephants to big places where you maybe want them to explore the area so that they'll know where everything is, you might want to target ones that are bolder. Whereas if you're moving them to a smaller place where you don't want them moving all over the place and you want them sticking in that small park, you might want to target the shyer individuals. And the essay that I use, which was how close they let me approach them with the car, is fairly easy to conduct before you decide who to choose to move. So that was kind of a brief overview of how by moving animals to a new place, you can really start looking at how a novel environment can influence social behavior, how it changes over time, and how individual variation fits into it. But most of my career, I actually worked on social insects. Here, an array of some of the species I've worked with. And the reason that I moved from wonderful elephants to these smaller, also wonderful animals is first because it's much easier to translocate ants than to translocate elephants. But also because there's this really interesting spatial constraint on the collective behavior of ants and many other social insects because they live inside nests. This is actually an example of a termite nest. And so really it lets you kind of look at spatial constraints on interactions, collective behavior in a very small scale that's easier to manipulate both in the field and in the lab. And we can just, kind of like what I told you with the elephants, we have spatial constraints interacting with individual variation in how different ants interact with their habitat and with their nest structure, and how that influences the social interactions. And with social insects, because the colony is the level at which natural selection acts, because most ants, the ant workers are sterile, and colonies produce more colonies. So we have the queen, she's the only one who lays eggs and produces workers, and she'll produce more queens that will fly off to create more colonies. And so natural selection acts at the level of this collective at the colony level. And all these ants, all these workers that we see, these sterile workers, they all interact and work on local rules. So there's no one individual that tells them what to do. Instead, we have these collective behaviors like collective foraging, nest building, and so on, that emerge from the local interactions that these workers have without any central control. And so there's a really important meaning to the social structure of these ants, because that's what determines what the collective will do. We also can then look in these systems how spatial constraints directly influence collective behavior. And because it's easier to manipulate these systems, we can start interrogating this feedback between the collective behavior and the spatial constraints. And again, just like I showed you with the elephants who create their trails, ants also create their foraging trails, but also they build their nests, or they decide which nest they're going to live in. So there could be feedback between how the animals decide, how the ants decide where to live, how to dig the nest, and how the nest then influences what they do collectively. And get this feedback. So I'll start by showing you why I think ant nests are so cool. And so this is a plaster cast of a nest that I've dug. You'll see some more later. And what I want to show you is what a nest looks like from the perspective of an ant. And so you can imagine this is basically plaster that filled the nest cavity, and then I dug it out. And now what we're going to do, this used to be the entrance up here. And what we're going to do is go into this entrance, I believe, and fly through the tunnels that the ants dug and kind of see what they see when they walk around the nest. So we're going to go down here, and there, get a feeling of what an ant nest looks like from the inside. So that's one, just to get you into the mood. And so the ants do lots of things inside the nests. They bring in their food, they store their food, they take care of their brood. They apparently watch TV and read books. The queen is protected down there. And so it's been something, something that a lot of people who study ants think about what the ants do underground, which we can't see. So it's a nice exercise in the beginning of a pandemic to do with your kid and think about what all the things are that happen down there. And it's also inspired a lot of thinking in human buildings and construction. And one of the things that I really like about thinking about buildings and architecture of ants is that it opens up opportunities to collaborate with other researchers from other fields. And so in one of the interdisciplinary collaborations I had with Steve, who's a social scientist, Yi, who's another ant researcher, and Ellen Penn, who's an architect, we brought together a group of researchers from our 3 different fields to discuss how we can use interdisciplinary approaches to uncover how architecture, the built environment, influences collective behaviors, all the way from ants through squirrels and of course humans. And just a brief kind of bit about one of the papers that I co-authored for that special issue is that is very timely because we're in the middle of the pandemic, is looking at how the built environment influences health, behavior, and disease. And I did this work with Nancy Wells, who's a social scientist at Cornell, and Andrea Jellick, who's an architect in Europe. And we talked about the parallels between how humans use built environment and animals use the built environment to prevent infectious disease. So for example, animals will use, birds will use bay leaf, and ants and bees will use resin to line their nests to prevent the growth of harmful microbes. And we use different types of paints in our houses and in hospitals that can be cleaned more. And so the materials that we choose in our buildings can influence whether or not we'll get disease spread. And similarly, we can use space, the built environment, to contain infectious disease. We all know about quarantine these days. Animals do that too. They'll remove sick or dead individuals from their nests to make sure that whatever killed them doesn't spread to others. And so going back to this scheme that I showed you, I want to start digging into this idea that spatial constraints influence social interactions and in combination with individual variation. And a lot of this work I started doing in the lab with Deborah Gordon as a postdoc, and then in my own lab later on. And so one of the things that I did, one of the first times I went with Deborah to the field was, I really wanted to see what the ants were doing inside the nest. So what I did was, I excavated the ceiling of the entrance chamber. So this is the first chamber that the ants enter. This used to be their nest entrance. These are tunnels that lead further down into the nest. And what you have here is basically the floor of the entrance chamber covered with a Petri dish so that we can see what they're doing. And you can see that the ants are bringing seeds in and out. We can see that they're interacting with each other, and we can then quantify these interactions and see what happens to these interactions as they get more food and as the environment changes. And so just a quick, for the first thing that we asked was whether the number of interactions increases when there's more food. So the dark blue boxes are periods with high food because we gave them seeds on their trail. And then the light blue areas are when we didn't give them seeds, so when there's lower foraging. And you can see that there's a lot more interactions. The interaction rate increased dramatically when there's more food. So they really use these interactions to recruit to more food. So there's a functional meaning to these interactions in terms of the collective foraging of the colony. And I'll just note that, they have a very short period of time every day to forage because it gets really hot in the desert. So they wake up in the morning, start foraging, and then close down foraging as soon as it gets too hot and uncomfortable. So they need to get as much food as they can in that small period every day, and they don't want to stay there for too long because they might fly out. What we also found, because we were looking at these interactions inside these entrance chambers, was we could see where the interactions actually happened. And so if this was the structure of the entrance chamber that I showed you, here is where the interactions happen. So again, the green is the nest exit, what used to be just the only entrance hole. These triangles is where the tunnels lead deeper into the nest. And what you see on the right is a heat map of where interactions happen. So where there's more red, there are more interactions, and the blue areas have fewer interactions. And so you can see that we have these interaction hotspots forming near both the nest entrance and the entrances to the tunnels that lead deeper into the nest. And we see that across different colonies and different nests. There is variation across colonies in the structure of that nest entrance and of that entrance chamber. And therefore there is variation across colonies in where those interaction hotspots happen. And in the lab, I was able to track in more detail all the movements of the ants and see that these interaction hotspots indeed happen also near the entrances to these tubes that are basically equivalent to the tunnels of the ants in the field. And what I found was that ants that spend more, because I had the detailed trajectory data for all the ants in the lab setting, what I found was that ants that spend more time in these interaction hotspots end up having more interactions overall. And what this leads to is these interaction networks with a very skewed degree distribution. So degree is how many other individuals I interact with. And so what happens is that we have few individuals that interact with a lot of other individuals, and most individuals that interact with just few other individuals. So all these blue dots are the ants that have very few interactions, and the red individuals are the ones that have many interactions. And so you can see that there's only very few red ones and a lot of blue ones. And this kind of network structure is very common in airport and in air flight, air transportation networks, and not so much, for example, in road transportation networks. And it's common in other biological systems as well. And usually these skewed degree distributions, networks of these skewed degree distribution facilitate fast information flow. That also means that it facilitates fast disease spread. So there's some trade-off there, and we can talk about that later. I then went on to ask how does individual variation in movement behavior influences the interaction network structure. And this was easier to do in a simulation. So I basically made computer ants and had them walk around based on rules that are similar to what we, well, the produced trajectories that are similar to what we see in ants. And on the left here, you see that a simulation where there was no individual variation. Everyone just walked in a random walk. Whereas on the right, you see a situation where there was high individual variation. They still followed the random walk with bias, but they had different level of persistence with their turning angle. And again, I can go to the details later if people are interested. But the main point I want you to get from here is that here everyone is pretty much walking the same way. And on the right, we have some that are walking in squiggly patterns and some that are walking more straight. And so when you actually look at the interaction networks that comes out of these computer ants, we do see more skewed networks in the high individual variation situation and very uniform interaction networks in the when there is no individual variation. Again, because these are simulated ants, we can also see where these interactions happen. And it turns out that when there's no individual variation, the interactions can happen all over the place. Whereas when we have high individual variations, the interactions tend to happen close to the boundaries of the simulated nest. And so turns out that this influence of where interactions happen are really dependent on the borders and where they are. So here's what I just showed you. And in both these cases, there were boundaries. As you saw, when ants made it to the edge, they started walking along them. They didn't bounce off like billiard ball because that's not what ants do. We simulated them to stay for a while along the edges, just to simulate what actual ants do. But when you remove the boundaries and you let ants walk on a torus or a sphere, or let's review here on this infinity loop. We see that even with high individual variation, interactions can happen all over the place. So there's no longer a structure to where the interactions happen, which then influence the structure of the resulting network. And we get networks that are much more uniform as well. So this kind of shows how spatial constraints combine with individual variation and movement patterns to influence where social interactions happen and how they're distributed among individuals in the society. And so I want to move on to how all of this together combines to influence the collective behavior of humans. And to do this, I moved to a different ant species. So up until now, I was talking mostly about Pogonomyrmex barbatus, and these are Veramester and Rreai. And what you see here is a relocation of an ant nest from one nest site to another nest site. And so you see the ants carrying their brood, you see the alates that they're preparing for the upcoming mating flight. And this is one of the biggest waves I've seen, but they do this quite often. And so what happens when you go to the field is you see basically a Swiss cheese of ant nests, and some ant nests will have active ants there, but a lot of them will be empty. And then one day you'll come and nest that was active previously is no longer active, but you see a nest nearby that's all of a sudden active when it wasn't before. And this happens throughout the season, and sometimes you actually get to see the ants move. And so what this really nice system allows you to do is look at the behavior of the same colony in different environments, right? So they do the translocation for you. And because we already saw that their interactions are influenced by the structure of their nest, we can then ask, how do they behave in one nest site, and compare the behavior of the same colony, the same individuals in a different nest site that's presumably in a different structure. And the first study that I did this just looked at their behaviors and compared them in different nest sites, and found that indeed colonies had the same behavior when they were in the same in a particular nest site, but then when they moved to a different nest site, they changed their collective behavior. And one of the behaviors I looked at was just recruitment to food. So how quickly they find a piece of apple they give them. I then later, that was towards the end of my postdoc, I moved to Southern California. I was able to replicate this study. And then this time, instead of just comparing their collective behavior outside and looking at how it changes as they move across nest site, as soon as a colony, I quantified their recruitment behavior to food. And then as soon as they moved to a new nest site, I went and I poured down plaster into their nest to create a cast. And so I created a cast of these nests where I also had quantified before their collective recruitment to food. And I was able to start relating the speed of recruitment of ants to food with the structure of their nest. And so I digitized these casts and turned them into networks because I like networks. In this case, each node is a chamber. So up until now, I've been showing networks in which nodes were individuals, but here nodes are nest chambers and edges are tunnels. And this is just another way to show it spread out so it's not easy to look at. And I use these networks to quantify different features of the nest. And particularly, I was interested in this entrance chamber that I've already talked with you a lot about, and how connected it is. So how many other chambers are connected to the entrance chamber. I asked about overall chamber connectivity, so just how much there are chambers each chamber is connected to. And then I was really interested in this feature of the network, which is called a cycle. So a cycle is basically a path that leads from one node all the way through other nodes and creates basically this closed loop. And the reason I was interested in this was because these cycles basically provide a measure of network robustness. Because you can imagine that if an ant tries to go from this chamber to this chamber, it has one path here that it can pass through. But if something breaks, a tunnel collapses, it still has another option, another way to get to that chamber. So the more redundancy of paths, one can think there might be more robustness of ways to get from one place to another. There's obviously also the trade-off of if you have too many of these, the structures might collapse because they are broken. So I was curious to see how ants navigate this trade-off between having these cycles that facilitate rapid movement and robustness of the structure compared to the lack of structural robustness there that they use. And so what I found was that recruitment speed increased with both chamber connectivity and this network robustness. And so here you have on the Y, the recruitment speed, so basically how quickly ants arrive to a piece of apple. And in this one, you have the degree, which is basically the number of other chambers that an entrance chamber is connected to. And you can see that the more chambers the entrance chamber is connected to, the faster the recruitment. Similarly, with overall connectivity, all the chambers, we see an increase with recruitment speed increases with connectivity, and then also with this meshiness. So the more cycles we have, the faster the recruitment. Interestingly, there was no relationship between recruitment speed and the volume of the chambers, which suggests that it's not just how many ants a nest can hold, but really how it is structured that influences the collective behavior of the colony. Then after this study, we started following up. So obviously, this lends itself to a lot of follow-ups, all the way from figuring out how these nests are structured in the first place to the feedback between the nest structure, the collective behavior, and the movement of where the ants decide to move and so on. And to start getting at what forms these structures in the first place, my two grad students, Eva and Sean, decided to ask how colony identity and species influence nest architecture. And they also looked at environmental features and asked if humidity and temperature influence the way the ants dig. And so they took these two species, Rreadings, which I was just telling you, and Pogonomyrmex californicus, so a different pogo species than the one from my postdoc, but in any case, and they put them in this elaborate experimental design where individuals from a source colony were divided into experimental groups, and each experimental group was placed in different environmental features, which were either cold or hot, dry or wet. And they put them in buckets, let them dig for a week, and then cast their evacuated the ants, so the least amount of ants were harmed in the process, poured down wax, created a cast, and quantified it, digitized and quantified it. And what we found was that when we look at the tunnel circumference, Rreadings dug bigger tunnel circumferences than Pogonomyrmex californicus. And we think the reason for this is because bear mice are slightly bigger in their bodies. So you can imagine if you're a slightly bigger ant, you need bigger, wider tunnels to be able to turn around. Their head size is the same. So it's not a matter of how big their mandibles are or the mandible reach they have. So it's really about the body size. Another thing they found was that the colonies differed substantially in the number of nests they dug with cycles, so that those cycles I showed you before, and also in how big of a nest they dug. So ants that came from the same colony, each one here on the x-axis is a different colony, really dug different sizes of nests. So they found both influence of colony identity and species on nest structure. Interestingly, they didn't find all that much influence of temperature and humidity. The only thing they did find was that temperature had, the only thing temperature influenced was the number of nests with cycles. So again, just showing you these cycles again. Here are two examples from the nests that Eva and Sean had. Here's a nest with a cycle, here's one without a cycle. And so we saw that there were a lot more cycles being dug when the temperatures were hot. And again, this could be because maybe for ventilation, maybe there's more airflow when there's cycles, maybe ants are more active and they move more, so they need more space to move around and not cause traffic jams. We don't know why this is, but this was the only influence we found in the lab of any environmental features that we tested. Another question that a postdoc in my lab, Julie, was asking similar time is, how does group size relate to nest structure? So there is more than 10,000 species of ants, and so this provides a beautiful opportunity to conduct comparative analysis and see if different species, compare the nest structures of different species and relate those nest structures with different features of those species. And Julie decided to focus on group size, on colony size, and asked whether species with larger colonies have nests with more chambers. So as species have bigger colonies, do they just add more chambers to their nests? Do they dig bigger chambers? And do they have more connections in their nests? And so to do this, she did a literature review where she found, we found all the nests in the literature and related to which species they are, found the size, the common size for each species colony, and looked at the relationship between, for example, colony size and the number of nests. And we found that there is a very strong correlation, positive correlation between the number of nests and the size of the colony. And you see different, each point is a different species and they're color-coded by ant subfamily. What we didn't find was a relationship with chamber size. So this is just a standardized measure of chamber size. So we're standardizing this to the size of the ant. And you see that there is no significant relation. We did not find a significant relationship between colony size and chamber size. And this makes a lot of sense if you think about unitary organisms, right? We know that mice and elephants have chambers, right? They have cells of similar size, but bigger animals will just have more of these cells than smaller animals. So just like unitary organisms, instead of growing the chambers, what ants do is they add chambers that are all of the same size. So it's like adding more cells rather than making your cells bigger. In terms of nest connectivity, again, we found that connectivity increased with colony size. We further asked, how does this increase happen? And compared 3 different models. We asked if nodes if chambers in a nest are added in a chain network versus a minimum spanning tree, which basically means that they every time there's a chamber, there's a split, versus a fully triangulated network, which means all nodes, all chambers are connected with all chambers, but without any tunnels crossing each other. And asked if an increase in size of the of the nest as a chain network relates to what we see in the observed nest, or whether the observed nests match more these minimum spanning trees or fully triangulated networks. And so I'll explain the figure to give you a better sense of what this is. The gray dots here are the observed nests that we had. So we can see the relationship between the connectivity of the nest and the number of nodes it has. these lines are what we see in the simulated networks. So if nodes or chambers are added as a chain, we will see a relationship between connectivity and number of nodes. It looks like this red line. If they're added like a minimum spanning tree with just like a few splits here and there, we will see something close to this orange line. And if ants build nests that are like completely connected, like these fully triangulated networks, we'll see the relationship between connectivity and number of nodes. It's like this blue line. And what you can see is that most of the observed points fall between the chain networks and the minimum spanning trees, with very small nests following a chain network and then getting closer and closer to minimum spanning tree. And I'll use this for a little kind of side note to mention this recent work I did with Pfefferman and Ms. Hobson, Matt Silk, and Dan Larimore, Sarai Shai, and Puck Rumbo, where we looked at these reference models or null models and how people compare different network structures to random. And we came from the point of view that a lot of network research, both in animal science and studying humans and other types of networks, because there's these interrelationships between the nodes, there's a strong reliance on creating a null distribution of what we might expect at random. But what random isn't always clear. And so we kind of developed this guide to what kind of reference models or null models people might be interested in comparing their observed networks to under different conditions. And we kind of classified them from the most random, right, the most shuffled, from the least shuffled, from just like small permutations that move identities between nodes and just do really small changes to the networks, so they're closest to the empirical observations, all the way to the most shuffled ones, which kind of think about the generative processes that create the networks, just like the ones I just showed you, for example, from Julie's work, where you think about what generated the network in the first place and see how that relates to your observed. And these require some knowledge about the mechanisms of your system, whereas these permuted ones don't require you to know a lot about what are the underlying mechanisms that form the interactions that you're looking at. And so basically, I kind of led you through this connection between spatial constraints and the visual variation, how they influence social interactions and collective behavior. And I showed you how spatial constraints influence collective behavior. With the feedback coming hopefully soon, we're starting to look at actually manipulating the structures that the ants are in and continuing the investigations there. As part of thinking about these problems, I bumped, one of my, Kelly Finn here, who's the picture on the top, hopefully I'm not obscuring her picture with mine, approached me a while ago, a few years ago, to invite me to a workshop about multilayer networks. And I knew nothing about these at the time. And so I was really intrigued and started, I went to that workshop, learned about them, and started thinking about them and talking about how you can use multilayer networks in animal behavior. And it occurred to me that they're a very good tool to create these links between the social settings that we're all interested in and the spatial settings in which these social interactions happen. And so along with Matt Silk and Mason Porter, we wrote a couple of reviews about how one can use multilayer networks to integrate social interactions in different situations, along with the spatial environment in which they occur. And so we're hoping to start using this framework. And one of the places we're starting to use this is in a recent NSF funded collaboration with Orange Spiegel to work on vultures. Now, I never thought I would work on birds because birds can fly, and that's an unfair advantage in my opinion. But when you have collaborators who really know the system well and can capture vultures and tag them and put GPS tags on them, we can start asking really interesting questions about how spatial features influence social interactions and how this differs across ecological situations. And so this is the framework we're using. We're looking at how the vultures interact in different areas, in different places. So for example, how they interact when they feed on carcasses. And so we can look at their interactions, for example, in a feeding situation. We can then look at their interactions when they're roosting at night in other locations. And we can look at their interactions when they're flying together. And we can compare these interactions that happen in different situations to networks where we all the situations are combined and see which situation influences the overall population structure and what kind of influence it has on the global structure. And so here's just an example of what the data looks like. Here's a vulture with a tag. Here's where the vultures are. And so, and here are some sample trajectories of vultures. And so when they fly together, it looks like this. These are two individuals that have small sections in which they fly together. And when they, and so we can use these co-flight sections to create networks of co-flight. And we can see individuals that are interacting together, sleeping at the same roost, which is this little polygon here, and create networks which depict who roosts with whom overnight. And we can take interactions in these different situations and look at the position of the different individuals in these networks. And what we see is that individuals vary in their position in different structures. So this is a plot that shows the degree of the rank of the degree of individuals. So again, degree is how many individuals each one is connected to. Darker colors means they interact with more individuals. And each of these rings is a different situation. So the inner ring is the co-flight situation. The second ring is in the roosting interactions. The third ring is the feeding interactions, or interactions during the day on the ground. And the last one is the aggregate. And each slice is an individual. And so what you can see here, for example, is that this individual, J15W, has a lot of interactions in the diurnal ground interactions. This individual, J36, has a lot of interactions in the nocturnal ground interactions, so while roosting. And then this individual has a lot of interactions when they fly. And so we can see that different individuals have different roles in different types of situations. And again, this relates back to the beginning of my talk of how we can use this information to inform wildlife management actions, because this is a population at high risk. They suffer poisonings, and there's a very intense wildlife management program for them. And so we can start seeing which individuals are more important in each situation and decide whether their management needs to vary by who they are. And I'll just bring us back before ending back to social insects, and kind of linking this idea that individuals do different things in different situation and how this relates to the collective system as a whole. And so when Nitika joined my lab, she just came out of RG's lab studying these beautiful wasps that I think you heard about a few weeks ago. And in the system, there's a single queen, so a single individual that lays eggs. And when she dies, a different individual will take her place. And this individual that takes her place is called a potential queen. And for decades, they've been looking for ways to predict who the potential queen is. And nothing about the potential queen indicated that she'll become it, not her behavior, not her physiology. And so with Nitika, for her PhD, looked at the interactions of these wasps in many different situations. And she had an interaction network for their interactions in space, how close they are each other, how much they aggress each other, how they share food, both solid food and liquid food through prophylaxis. And so what we did is we decided to look at the role of the different individuals in these different situations, but also in a multi-layer network that kind of combines everything together. And what we found was that this potential queen, this is again a similar plot that I just showed you, where each slice is an individual, and the potential queen here is highlighted in yellow. And what we found out was that only when we looked at the aggregate network, which was basically smushing all of the situations into one single network, we weren't able to distinguish the potential queen. We also weren't able to distinguish the potential queen just by looking at each of the layers separately. But when we created a multi-layer network, which takes into account the fact that the same individual appears in different situations and it takes that into consideration when creating and when calculating the out degree. We found that the PQ was finally, we were finally able to identify who the PQ was based on its interactions across all situations. And we found that her position in the multilayer network was significantly higher. So her out degree was higher, her strength was higher than all others, and greater than what we would expect at random in all the 5 colonies that we had. And so this just brings back the idea that multi-layer networks really can be helpful to examine links between behaviors in different situations and across spatial structures, which brings us back to this idea that spatial constraints, individual variation together influence social interactions to impact collective behaviors. And hopefully you got from this talk that spatial features and ecological situations influence animal movements that influence interactions and determine the emerging collective behaviors. I'd like to thank all the many collaborators, funding agencies, students, of course, in places where this work was conducted, and I'll be happy to take questions.
Okay, well, thank you, Noah, for a really fascinating talk. We have lots of time for some question and discussion. And at this point, I ask if you have a question, please type a question mark in the chat, and I'll relay that to Noah. And if you're on YouTube, please type your question into the chat there, and that will be relayed to the speaker. And as always, please, when you ask your question, identify yourself, give us your name and your affiliation so we can get to know each other. And we really encourage students and postdocs, etc., to ask questions today. So I'm going to start. We have one question in the chat, and that's Karsten.
Yes, hello. My name is Carsten Rreadings. I'm a researcher at the CNRS in Strasbourg, France, studying social organization in mammals. So I know very little about social insects, and I still struggle to understand social network analysis and so on. It was a great talk, a lot of information. And my question relates to one thing which just would like to be explained better, as I lack the experience. It's on the graph you showed with these different kind of networks in the vultures, where you had these different shadings of gray. I have the privilege to be a co-host. I can quickly show you which one I mean. I mean this here. So my question is, how repeatable is this? Because for me, I mean, of course, you put it nicely in color. It could be just a random structure. If it just randomly, you find something more or less gray. So is there somehow a check that this is really repeatable? But like when you said this individual always has a lot of interactions, especially on the ground.
Yeah. So this data is from the breeding season. And so we look at a network. We look at these 3 networks in these 3 different situations. So at this point, it's early. I was excited to start showing you some early data. So we don't have, well, for this particular one, we don't have repeatability yet. We can calculate it. But given the interactions that happen all at the same time, right? So the data for this annular plot was taken all from the same time for the different situations. So let's say from December to May, we looked at all these individuals, looked at all their interactions when they were feeding, or on the ground during the day, presumably feeding, all their interactions at night, presumably when they were roosting, and all their interactions in flight. And so for each individual, it's the same time period that we're looking at. And so the color coding is the rank. It isn't the absolute number. It isn't the absolute out degree, but it's ranked among all the others and scale to the other players as well. So we can still say that an individual in this time period is more is more interactive in one situation compared to other situations, because we're comparing different situations within the same time period. Does that answer your question? I mean, and then you're asking whether it's repeatable across years or across seasons. So that we don't have that. That's kind of the next paper, as relating this to age and, sex and so on, and starting to look at whether there is repeatability in movement patterns and therefore in interactions. So that's the next step.
Okay, so you cannot be sure that that's an individual trait of this individual, and that one will be the next year again, especially on the ground, and the other one especially co-flight and so on.
No, and it's very possible. It's very possible that it even changes within a year. So this is focused on the breeding season. It could be that the non-breeding season gives us a very different pattern.
Okay, thank you.
Okay, so our next question comes from Michael Taborski.
Michael Taborski from University of Bern in Switzerland. Thank you very much, Noah, for this interesting talk. I have a question regarding recruitment. I mean, you mentioned that more or less in passing, but I think this is a very interesting aspect regarding the type and amount of information that is transferred by these interactions. I mean, you mentioned when you opened this entrance chamber of the ant nest, that when you increase the food that is available, they have more interactions, and you mentioned in passing that this may also involve recruitment. And I just would like to know whether and to which extent which information is passed and how this recruitment really works.
Yeah. So in the polynomics verbatis system, we know a lot because Deborah has been studying them for decades. So we actually do know what information is being passed. In that species, they don't really lay. So you might be thinking about trailing ants. You don't really lay a very strong kind of chemical trail to lead other ants to, but instead, what happens is the ants are queuing in on the interaction rate. So as the rate of interaction increases, it increases their likelihood to leave the nest. And the way we can actually test this, and people have done this in the lab, is we can take a forager ant, extract the cuticular carbohydrates off its body, and put them on beads, and put the beads into the nest at different rates. And it turns out that if you change the rate at which you drop beads in, it changes the rate at which ants comes out. And another thing is we know what kind of smells they are interested in. So collaboration I did with previous postdoc with Deborah was we compared the interactions and the response to beads that had different types of smells on them. So we had beads that smelled like foragers, beads that smelled like seeds, and then we had beads that smelled like both foragers and seeds. And it turns out that it's the smell of the forager and seed, and then we had beads that didn't smell like foragers, but some generic other ant. And then the smell that they responded best to and interacted most with was the seed plus ant. So just seed smell isn't enough, just forager smell isn't enough, but it's the combination of the hydrocarbons that surround the foragers and the scent in the outside of the seed that really triggers this response and this recruitment to food. So if I'm an ant kind of waiting in the infant chamber, deciding whether or not I'm going to leave the nest, I'm basically sitting there and counting interactions somehow. And when the rate increases, when enough successful foragers, so foragers with seeds, come in and I bump into enough of them at a certain amount of time, I will leave the nest. We also saw that that animal behavior people goes into a lot more depth on other aspects, but one of the things it talks about, one of the things it talks about is how the colony controls how many ants are in the entrance chamber. And that kind of changes over time in response to kind of longer term disruptions in food and longer term return rate of successful foragers. So they kind of not only regulate the interactions, not only does each individual respond to the rate of interactions, the colony as a whole also regulates how many ants are available to interact with these returning foragers. So that's kind of what happens in those desert ants. Does that answer your question?
Yes, more or less, but I would still like to know, I mean, regarding the information that or the change in their behavioral responses to these different smells. Is there any indication that there is more than just an enhancement of exploratory behavior? Is there more information than just there is something out there?
So they leave the nest and start foraging in the same direction that everyone else is foraging. They forage in a different direction every day. And so what happens early in the morning, there's another task group of patrollers that they leave at dawn, and they kind of scout the area, and based on whether or not they interact with other ants from other colonies, whether or not they find food, they determine which direction the ants will forage that day. And so they return and basically, so I think there's a little bit of the pheromone left on just the mound itself. So it's a very, very short one that just kind of gives general direction. And then ants will just start zooming in that general direction, and they'll just keep walking until they reach the food and they fan out and they start collecting the seeds and then return on the same trunk trail. So they form a trunk trail, but it's not, the trunk trail itself is not completely covered with pheromone. It's just they have the general direction, and then they form these like foraging fans that go off from and the reason is because they don't feed at a particular place. It's not like they're all going to some dead animal or some I don't know, like a fruit tree or something like that. Their seeds are kind of dispersed in an area. They're dispersed, and like the rains will flood and kind of make the seeds even disperse further. So they're kind of homogeneous, but like within a patch, they're kind of homogeneously spread out. So you kind of really have to fan and find the seeds, and it's not like just one place to see. With the Verrumosa, I mean, I've seen some Verrumosa species where they will go to a particular bush and like start picking things from it. And so again, I don't know. I don't think we know whether or not they actually have a pheromone trail all the way there or not.
But the interaction itself is just enhancing the activity, more or less. And then
Yeah. The interaction is basically just activate an ant to leave the nest, and it'll just go in the direction that it needs to go.
Yeah. Thank you very much. Okay.
Sure.
Okay, our next question is from David Wood.
Hi, thanks for a super fascinating talk. I'm David. I'm a graduate student at Yale working with Eduardo Fernandez-Duque. And I'm curious about how you calculated this aggregate score in the multi-layer network analyses, if that's just like the average ranking or the average degree for the different networks, or if you were able to weight certain networks above others so that, some networks may have like more valuable interactions than others.
Yeah. And both the studies that I showed you, we just did a linear sum. So you're right that we could decide that some interactions are more important than others, and maybe they would get a higher weight. But what we did is we basically took each of these layers and just took the edge list of them and just concatenated them. So basically, if you have an edge between two individuals, it'll also appear in the aggregate, and if there's an edge in the three situations, it will become a weight of 3 for that edge in the aggregate. So we just did a linear sum. You're right. I mean, this is something we discussed in the review with Kelly's lead author on. People, depending on the system, depending on your question, you might want to give different interactions different weights. Maybe some interactions are more important for the relationship that you end up looking at. So I kind of view these aggregates as the culmination of all these interactions in different situations. It's basically the relationship that is built in all these interactions that happen in different contexts. And that would really depend on the system that you're studying and the questions you're asking. And similar with also with the multi-layer network, when we did that, we also treated them all equally, but we could also decide to give a certain weight to a different weight to others.
Thanks.
Okay, next is Susan Perry.
Hello, Noah. It's funny, I see my UCLA colleagues more in this fine seminar than I do actually in LA. We should change that. But anyway, I'm really interested in the multi-layered network approach that you are talking about for some problems I'm about to try to tackle in my own Capuchin data set. So I was wondering if you could tell me more about the format of the data that goes into these. Like, does it have to be binary or can it be continuous? Basically, what I'm trying to do is I have a data set with various kinds of social interactions. Some of them are, percent time spent together. I have a continuous measure of relationship quality, personality score varying from 1 to 5 from questionnaire data, and various other things. And I'd like to use these perhaps collectively to predict a distribution of particular behavioral traits that I think are socially learned, possibly. Furthermore, I'd like to compare time periods to see whether changes in these predictor matrices are associated with changes in the patterning of the cultural traits. And so far, I mean, I haven't done an exhaustive search of literature, but I'm having trouble finding a social network approach that's really going to accomplish all of these things. For example, these SIOM seem to require binary rather than continuous measures. And I figured you're much more of an expert than I am. Maybe you know the answer to this.
Well, I don't know that the answer exists.
Okay, that's what I was afraid of.
It might be possible that a measure needs to, like someone needs to develop a measure that would work for your questions. It sounds like multi-layer networks could work. We have one of the biggest experts in the world on campus in that, so you can reach out to Mason about whether measures exist and whether he's willing to sit with you and develop some. So that's the one thing I would say is that, this whole framework of multi-layer networks is fairly, it existed conceptually, but bringing it together into something cohesive with a formalism that kind of aggregates everything happened around 2014. And a lot of measures are still being developed and come out every, I wouldn't say every day, but frequently. But the nice thing about this framework is, the nice thing about it is that you can combine net interactions that happen in different situations and also happen in different times. With times, it's always hard because, time is continuous, but we end up describing it always in the end as discrete things in networks. Even there, you could do time ordered stuff, but still there's some unit of time that you're going to aggregate things by, because that's how our brain works. So in that,
that too, I've looked at these NVDA, but it seems to me like there isn't a good way of dealing with the problem we always have in naturalistic studies, where you have observation gaps. And so in some sectors of the data, you're much more likely to get the time wrong than in other sectors. And I guess there's maybe, or theoretically could be a Bayesian solution to that, but I don't know how to do that.
Well, I mean, I think you need to take into account the constraints, but I will say, so there are things, when you look at in the multi-layer network world, things like reducibility and kind of how much information you need to get some of the measures that you have. So there are ways to look at kind of, if I add more interactions and more layers, am I also adding more information, or does it saturate at some point? So there's tools to look at that. There's tools to look at, so a lot of the stuff we've been doing is kind of looking at individual, kind of the equivalent of centrality in the multi-layer network world. It's called versatility, and how that relates to centralities in each layer. So kind of looking more at the individual roles. But there's also like Mason is an expert, for example, in finding cluster, like how you define clusters, whether you consider multiple situations, multiple time points, both of them at the same time, and whether you can identify kind of clusters of and if you're combining time and situation, it would be like a time situation with a time situation combined cluster where things might happen. So there's definitely tools out there, and definitely like opportunities to develop ones and tailor them. And I think one of the things that's kind of I would say the strength in networks is this reliance on reference models, and what is it that you'd expect if there wasn't a structure, so you have a question and a hypothesis about what you think is there, and you can go and shuffle your network in a way that breaks the relationships that you think are there, and then you can start comparing what you did to what you saw to these random or reference models, and you can do them in monolayer networks, the regular networks that we look at, but also in the multi-layer network framework. So I think a lot of it requires kind of sitting down and really thinking about what the hypotheses are and what the questions are, and what are the relationships that you're interested in kind of interrogating and breaking in your null reference models, and then to start seeing if what happens when you break them, does your observed still match what you see? And so maybe those weren't the relationships that were really important and something else is. So I think it's kind of, there's many layers. There's like the, sure, you can plug your networks as they are. So we use, one software that was developed for analyzing multilayer network is called MuxViz. It requires your data in a certain,
Put that in this.
yeah, sure, I can put it in the chat. It's called Mux, hopefully I get it right, MuxViz. We should be able to Google that and find it. Again, it requires a particular structure to your network, and again, it's, while it can help you visualize them and get the versatility and the reducibility and some of the other measures, it's not everything. It's not. The last time I used it, it was still only for multiplex networks. So multiplex are a subset of multilayer, in which all the nodes appear in all the layers. So this is good for social situations where you have all the individuals in each and every one of the situations, but it's not so good for like linking the social and the spatial, where you have different types of nodes. And so measures for those types of networks are not there yet, at least last time I checked. Our packages are starting to come out for multi-layers. And then I think a lot of it, because it's still kind of a developing field, a lot of it is just people write their own code to do the analysis and to create the measures. But yeah, I'm happy to talk more offline if you want.
That'd be great. Yeah, let's do that. Thanks.
Okay, any other questions, please throw a question mark in the chat. In the meantime, no, I have one about, I have a series of small questions I was going to say, but I'll throw one in here. And I apologize if I missed this. In one of your networks, you had different colored dots for different individuals, and the red individuals caught my eyes. And do you know anything about those individuals? What makes them so important?
Yeah, so that's the ANT network. It's an earlier one where I basically, so yes, the ones that are highly, so yes, I kind of know why they're red. The reason, so the red ones have more interactions than the blue ones, right? And so what happens is they spend a lot of time in the interaction hotspots. They just hang out near the entrance and just bump into a lot of other individuals. Why do they stay there? I don't know. I did find in a little study I did that I never ended up publishing, that there is actual repeatability. And so individuals that tend to interact more will do so over a few days. Maybe because there's some site fidelity that they stay in a certain, that they like a certain place for a few days, maybe something about, I don't know, their metabolism, hunger level. I don't know what makes them stay in those areas that make the bumping strong, bumping into other ants. But after a week, this kind of repeatability breaks down. So they'll be consistent in the number of interactions for, I think it was like 3 or 4 days, up to 3 or 4 days, but then after 7 days, the repeatability went down. So I think ants are not as smart as we think they are. Collectively, we have these really smart emerging patterns, but in the end, it's based on, just where they are and where they bump into. That same paper, we also looked at whether they kind of preferentially go to those places, like whether ants prefer to go to the places where they will bump into other ants. And we didn't see any evidence for that. It's like they walk around, and if they bump into someone, they bump into someone. So that's why I'm saying that the structure of that's kind of what got me into thinking about, wait, if it's all about where they walk, who they bump into and how much they bump into others, in the end is determined by where they are and what the structure of this chamber looks like, then the space and the spatial constraints have a huge impact on what will end up happening down the line for the collective. So that's kind of what led me down to this long path of looking at the effect of spatial constraints in architecture on collective behavior.
So along those lines, I think it was in the Miller et al. Evolution paper you were talking about nest structure and group size and things like that. But I don't remember, and I apologize again if I missed it. Did you look at soil composition? Because I would imagine
No.
like what they can build.
Yeah.
Yeah.
And people have looked, people have looked at a little bit of the effect. I think I just heard, there's some studies from, I believe, Flavio Rosas's lab that I think it's coming, either came out recently or coming out soon, that looked at leafcutter ants in different soils, different environments. There's a little bit of a bias, people study ants and sands that are easier to dig. So there's that little bias in the literature. So there are, some, and we did restrict only to ants who dig in soils and to nests that we could quantify and are not like spaghetti like, we couldn't quantify from the data that we had, from the pictures that we had. But yeah, I think it has, the soil and its structure can have a big impact. So when I did, all those castings of the Vermeer that were moving between nests and all that, I had more than 70 casts, and I had so many rocks in there. They were big, and then they'll hit a rock, and so a chamber will form there. And the size of the chamber could be determined by how much it takes them to go around the rock. So I think definitely there's an influence of and I had a little undergrad project that started but never ended, kind of looking at how the distribution of rocks in the sand influences the structure. So that might, maybe we'll pick it up again someday. It was pretty straightforward. When you have rocks, you get stuck, so it ended up not being as intriguing. But yeah, it should definitely, it goes back to the ecology in which they live and how that influences what they can make.
I have more questions, but I'm going to defer now I'll go to Karsten next, please.
Yes, thank you for having the opportunity to ask a second question. It's a very basic question. And sometimes I feel embarrassed to ask them, but when I ask my students, they say it was actually interesting for them as well. And with this, I would also encourage the students and younger scientists, if there's something not clear, just ask, because there are other brains in the audience like mine. It also doesn't understand everything. So my very basic question is, your main study species were Meso and Race. Can you tell us more about this? And did you choose it specifically for your research question, or is this a species that's in every lab where people work with ants? Or why do you study exactly that species out of 10,000 species of ants?
Yeah, choosing your ant species is a very good question. So the Vermeer, I really like, I don't do much work with them in the lab. We did the Sean's and Eva's data, we brought some in the lab, but really, I like their behavior, that they do this their relocation behavior is, I wouldn't say unique, because many other ants do this relocation behavior. But it really is kind of very large scale rotation. A lot of the nest relocation studies have been in these teeny tiny ants, these Temnothorax ants that live under rocks in very small cavities and are really easy to study in the lab. And there's many labs that study them. But really the Vermeer and Rread give you kind of an opportunity to study these nest relocations in a natural setting. So they're not super abundant. They're actually, there's some populations, like the population that's closest to us here now in California is a really nice one, but it's still a couple of hours away. So it's a combination of, yes, they're here in Southern California, and also where I was in as a postdoc. And yes, they are big ants with big colonies, and also yes, they have a very, very nice natural history that allowed me to ask the questions that I was interested in, which was, I started this a little bit because I wanted to see if colonies have consistent behaviors. And so that 2012 paper was basically looking at colonies repeatedly, and then because of their natural history and the moving, I could ask, like, does this behavior change across different nests, and is there a kind of colony personality that's inherent to the ants, or is there something that changes because of the environment in which they are? So it kind of was a combination of, these were ants that I could study, they're big, they're easy to see during the and it's not like you go and they're always there. They're very finicky. They will only be active like from around 7:30 until we start sweating. So they don't like the heat either. So it's comfortable in that sense, that you never have to sweat in this field, but it also is very uncomfortable because you have, like, sometimes you only have 2 or 3 hours, and we need to wait till tomorrow so we can continue working on them. So yeah, I think it's just a combination. And then for, so yeah, it's a combination of availability and natural history, because there's other ant species. A lot of the work I didn't talk about that a lot of undergrads do in my lab is with Argentine ants. And that's because they're everywhere and we can just step outside the building and collect as many as we want, and we don't have to maintain them in the lab. So I prefer not having to maintain them in a lab, because I don't want lab maintenance to somehow mess with their behavior and change it. I'd rather like bring the ants in and either release them or keep them for other reasons, and kind of maintain that natural. But sometimes we do bring them in for a week or so. So yeah, it's a combination of availability and questions and fitting the species to the question.
Okay, thank you.
For the next question, I'll just echo Carson's comment. I think it's important for the students to know that every time I ask a question, I'm terrified. It's important for them to realize this and to go ahead and do it. That was a great question. Why did you study that animal? That's a great question. So that out there, now that it's out there for the world to see, Sean, you're next.
Great, hey, thank you. Hi, Noah. I really enjoyed the talk, great stuff. It made me think a lot. So I fired you an email message about something I'd like to talk with you about later in more detail. So just to introduce myself, I'm a professor at Drexel University in Philadelphia. I'm sitting at home, locked down in Swarthmore. And I'm interested in social insects. A lot of my work right now is on brain plasticity and brain evolution. I'm also interested in thermal physiology. But I have a grad student who's actually going to defend in a couple of days, interestingly, her PhD, and working on division of labor in paper wasps, which is something that I've been working on for a long time. And so this student, Katie Fioca, we just put out a short paper where we used social network analyses as a tool for analyzing dominance interactions and behavior and division of labor in her paper wasps. And I guess it's not really so much of a question that I have, it's a little bit of a complaint and a rant, and I'd love to hear, Noah, your thoughts on this, and maybe anyone else in the audience, whether if you've dabbled in using social network statistics. This is a frustration that's come up for me, and I'm curious whether folks have found ways around this. This is the deal, right? I mean, so we can build social networks, and I think they're pretty meaningful. I think they're a valuable tool looking at the interactions among our animals. And what I've discovered is there's a whole set of really interesting networks that range in sophistication, or a range of stats rather, for describing and quantifying characteristics of social networks. And what I've come to realize is that the majority of these don't apply to animal behavior interactions. So we've ended up using these really kind of doltish, very basic descriptive metrics of network structure, and I think that they've been valuable, and I think they've yielded some insights. But it seems to me like a lot of the social network statistics have been developed for human information transfer. So if you can think meaningfully about and have a reason to expect that me sending a friend or a like or whatever to somebody on Facebook, and then the number of times that they come back to me and the number of times that transfers out to other people is meaningful, right? But in a lot of cases in animal behavior, it's not actually meaningful to talk about the information in that way, because we don't really understand whether or how information is transmitting through individuals. So if a wasp has a dominance interaction with another individual, and then the individuals that she's having a dominance interaction with are linking to others, it's not really clear whether or how the interaction between two individuals transfers and ramifies through the network. And So this is just something that I found kind of limiting and frustrating, and I'm wondering if there, people have developed or are interested in developing, tools where we can go beyond simply describing how often individuals interact with each other and just kind of leaving it at that. So I hope that made at least a modicum of sense. Anyway, something that I found really limiting in trying to use social networks as a tool.
Yeah, I mean, I think a lot of the transmission type measures and transmission type analysis that I see in animal social networks is to investigate disease transmission. So in those cases, you can see that disease is transmitted. You can also, simulate disease you can simulate microbes and kind of start looking at how microbes mix in a society, whether they are disease inducing or not. So I think, first of all, yes, you always have to fit the measure and the, tool that you use to the question you ask. That applies to anything, not just to networks. And yes, there are way more tools out there than you will ever use for any particular study. So I don't think that networks are different in any way from any other statistical tool or other even just measurement tool, right? I won't use a scale that in the lab to weigh an elephant. So I think there's a lot. You really need to consider them as a research tool and a statistical tool and use whatever fits your question, not say, oh, what a cool measure, let's find a question that fits this measure, because you might not. And also that's not how we do science. I mean, it's kind of a little bit of backwards way of thinking about it. So I will say that, there are flow type measures that do fit questions about disease transmission. I think that in some cases, information flow could be relevant. I mean, if there's social learning and there are systems, I think Lauren mentioned that you'll see Lucy's talk at some point next quarter or next semester. Over there's in her work with birds, you see information transmission through social information, which side of you go for red or blue, and that's socially transmitted. So there could be in systems where you have learning and social learning, you might be able to distinguish between individual learning versus social learning. And the WASP might have it, but we might not have a way to see that. And so I think that as long as we can say, okay, this WASP learned from this WASP, then we might not be able to do that. But like in honeybees, we know that they have a dance, right? So we can see, oh, this bee danced, this bee watched the dance. Now this honeybee is going to forage, and you can start forming, interactions with information flow in that system. So I really think it's a matter of how much we can interrogate the system and see the world from the point of view of the animals. And it's not always possible because, we don't have all the tools to do that in all the systems. So does that kind of answer? I feel like it's not, I won't have an answer for you of like, this is how you're going to study how wasps talk to each other and what, like, I can tell you that when I was a PhD student, I did have these vivid dreams when I was in Africa, and I was always, one of the dreams I had a questionnaire with one of my elephants, and like we sat together and we had tea and I asked questions, and it was lovely, and I got to know all about that particular elephant, but that was a dream. And so that does not happen. And so, until we can like sit with our wasps and ants and ask them what we want, we have to keep on figuring out ways to extract that information from them.
Okay, so we have another question coming from Jingyu.
Hello, I'm Jingyu I'm a PhD student from the University of Strasbourg in France. So actually, I have a question about the really cool 3D model you showed at the beginning with the ants nest. Could you quickly explain the method you use to create these figures? Not figures, this model. Do you use
the movie of flying into the nest?
Yeah. So do you use a destructive way or non-destructive way?
Yeah. So the way I create the cast is I waited for the ants to evacuate the nest, and then I poured plaster down the nest and then waited for it to dry. And then I dug the, so it is destructive. And then I dug this, dug the soil and the rocks, brought it into the lab, cleaned it up from all the dirt. And so basically the plaster that, flowed into the soil ends up giving me the structure of the nest. And so then in the lab, I digitized it. Are you interested in like the digitizing process or just like the getting the nest cast? I wasn't sure.
Oh no, I think I understand the
Okay,
process.
yeah. So it is destructive. It is destructive, but because these ants vacate the nest, I didn't kill any ants, so I just killed their nest. And then we cover up, once we create this hole, we cover it up and hope that someone will maybe dig there again one day. But yeah, it does destruct a nest, but luckily there's still lots and lots of other nests and we can dig them up again.
Yeah. Okay, thank you.
So staying in the spirit of students, I'm gonna jump over to Eduardo and come back to you. Linda Lani. Go ahead, please.
Okay, hi everyone. Thank you, Noah, for the very beautiful talk. I really enjoyed it. So my name is Linda Makoya, and I'm a PhD student with Carsten Schraden and Neville Pide. And I'm working on trying to understand the cost and benefits of solitary living. So my question is, I saw in the beginning of your talk, you were you focused a lot, you were focusing on elephants, and then you did the whole shift from the elephants to the ants. So how was it in your studies when you did that whole shift, like from working with such a big animal to quite very minuscule one?
It made life easier. So first of all, it made life much easier. I always wanted to study elephants, and I felt like once I did that, I could move on. I feel like, in terms of the questions that I'm asking and the tools that the analysis tools that I'm using, I didn't have to change a whole lot. I always say, once they're in a matrix that you can turn into a network, they're all the same, whether they're elephants or ants. I think it just made the, actual ability to do experiments and kind of the type, a little bit changing slightly the types of questions that I can ask and the type of manipulations I can do with the system obviously changed. But the broader questions of, how the environment influences social interactions, how personality or how individual variation plays into the social structure. These are questions we can ask across systems. And these are kind of questions that I've been asking in many systems. So I think in that sense, I didn't have to make such a big shift. The size shift is more dramatic than the shift in the questions and the analysis tools that I had to make, if that makes sense.
Yeah, it does. Thank you so much.
Okay, Eduardo.
Thank you. I'm Eduardo Fernandez Duque at Yale University. Thank you, Noah, for a wonderful talk. And I'll have to refrain myself. We have a saying in Argentina, but I don't think it's appropriate for this audience. We have minors linking the ant and the elephant. So I'll share with you. It's funny, but I don't feel comfortable sharing it. Enough said. So I really want to go back to what you said a few minutes ago, because connecting to Carsten's very humble, very open comment on how we sometimes just feel like we don't know a thing about ants or elephants or any of the wonderful systems that we hear about are fine. And I think it's so important for our younger colleagues and our students. You made a comment in answering, I think was Susan's, or I don't know what's the question. She said, it's just a tool, the issue of network. Oh, it was Sean, right. What do we get out of social networks? What we get out of what people may be doing, developing mathematical tools to do social networks in humans. That's a challenge we face all the time. And we're jumping from ants to elephants, not to mention viruses and bacteria. And I want to go back to what we were discussing last week. I mean, tell my young colleagues and the students, you are the expert on that single ant species out of 10,000. And so how much you can get how much you can get from sophisticated mathematical tools that summarize social interactions in humans, which, for example, for the most part, pretty much everything we do as humans is very close to being bidimensional. When you think about birds and fish, I mean, so many animals move much more in a three-dimensional space than we do, right? And suddenly we're summarizing everything humans do in a nice drawing, colorful, and very powerful, but it's such a simplistic view. And so if we can just remind ourselves not to forget that these are tools, and I link it back to what we said last week, and then we need the expert judgment. We need the Noa who has been looking at this single species of ant for years, or we need Michael who's been looking at fish, and there's no math, there's no tool developed for looking at humans that can help us make sense of what we actually observed. So thank you for saying, and I just wanted to be sure that it doesn't go unnoticed, is just one more tool to produce useful summaries of extremely complicated interactions between organisms that I don't think we'll ever know. I mean, you said sometimes we don't get to know how the animals perceive the world. I think that we never get to know how they perceive the world. With some talks are more complicated than others, but that's all we can do. But thank you for bringing that up. Very, very important, I think.
I mean, I think if we, I hope we will never run out of work, because that's what we do. I mean, even people who study people don't understand people all the way through. So, I mean, it's always, you know, you ask questions and you collect evidence to either support your hypotheses or maybe sometimes you cannot do that. And I think in general, yeah, I feel like you had a comment, and so I don't really need to respond to it. It's more of a just kind of extending it a little bit. But I do think that that's what we're constantly doing. We're trying to figure out how the world works, and it's a complicated place. And well, maybe physics figured out some things like optics and stuff like that. Maybe we know how light works, but maybe not. Maybe they'll again have measurements tools that all of a sudden discover some other particles that they haven't been able to see so far. So in that sense, yeah, the better your tools are, the more information you can gather about your system and the more information you can extract from it. So.
Okay, so our next question comes from Clara.
Hi, thanks for the talk. Clara B. Jones, retired social evolution. I use a major transitions approach. I noticed that you have a paper coming out in Evolution. Can you explain how the work you presented today is related to evolution?
Yeah, so that particular work was a meta-analysis that compared nest structures across different species of ants. I didn't show, I tried to fit so much in. So for each paper I showed, I showed like a snippet and just a couple of figures, but we also looked, we did this analysis in a phylogenetically controlled way to ask whether it's, something inherent about the size of the colony, or whether there is also an evolutionary signature there, and whether maybe closely related species tend to have similar patterns of, how nest structure, the shape of the nest, connectivity, number of all the things we measured about the nest, or whether it is related to just how many ants are in a colony. In that sense, we actually found, I can probably pull up that figure, the tree figure that maps all our different measures onto the tree, and we actually didn't find a strong phylogenetic signature in that analysis. So basically showing that it's the size of the colony, which isn't necessarily restricted by the evolutionary tree, that determines the structure of the nest, or that relates to the structure of the nest, rather than some kind of evolutionary origin of all this clade is going to have highly connected networks, and all this clade is going to have lots and lots of chambers, rather than it's kind of spread out across the evolutionary tree of ants. So basically, because this was meta-analysis, I think, more than 40 species that allowed us to do this in phylogenetically controlled way and ask also other questions about whether it's there's an evolutionary origin to this structure that we found. That's kind of the relation with evolution there. Does that make sense? Yeah.
Well, yeah, I want to ask, does that mean that you can show that the data you presented today, let's say for one of the, let's say for the social insect, that those data are repeatable?
So we measured many nests for each species. Is that what? So we have multiple measures.
I mean,
nests.
if it's going to be subject to selection, then you have to have the same patterns over and over again so that they can be exposed to selection.
Yeah. So the work, Sean and Eva's work that really showed that there are consistent behaviors in the colony. So suggests that there could be selection working on these structures. So we view these nests as extended phenotypes, right? They're a phenotype that's constructed from the behavior of the ants. And so presumably it's selected on. And so the lab study that we did that compared these two different species, and we saw very strong signatures of species. So one species tended to dig bigger tunnels than the other species. There could be selective pressure there, because if you dig tunnels that are too small, you won't be able to turn around. If you dig tunnels that are too big, things might start collapsing, or maybe you'll get other animals coming in. Maybe it's harder to protect. I know we could come up with a lot of stories and hypotheses for why that is. But also the fact that we saw when we took different groups of ants from the same colony, and they ended up with very similar structures. Like they ended up digging there were colonies that dug bigger nests. And these are 4 independent groups that came from the same colony, and they all dug bigger nests when you compare it to 4 groups that came from a different colony. Also suggests that there's some inherent colony identity signature in the structures that they dig. And again, this is 2 different species. So there is, in that sense, there is this repeatability of, yes, we're taking individuals from the same colony, different groups of individuals from the same colony, and they end up digging similar structures, in the measures that we looked at. So there is space for natural selection to act on this, because these structures emerge from what the ants do. Hopefully that addresses your question. And I think part of that, that study with Mountain Visions is really does suggest that it's more than just the environment and like how hot it is out there, or how humid, which there's been other studies that show that if you really change the humidity of the soil, you can get different structures. And we talked about it earlier, right? If you have rocks, you're going to be constrained by the rocks. But if you remove all that and just give them as much sand as they need with no interruptions, then there are certain characteristic structures that colonies will dig out. And there's other studies, not from my lab, that look at, for example, the size composition of a group and how that influences the structure of the nest. And there's a really nice signature there where groups of smaller individuals will dig different structures than groups of bigger individuals, and the size of the individual is genetically determined and determined by other developmental aspects. So yeah.
Okay, thanks.
Okay, so just an organizational thing. We have 3 questions left, and I think then we'll give you a break because you have a series of meetings with some of the people from the organizing labs. So I have 2 questions in the chat here and then 1 from YouTube. And so the next question comes from Adriana.
Hi, Noa. Thanks for your nice talk. I really enjoyed it. I'm Adriana Maldonado Chaparro from Universidad de Rosario in Colombia. And I have a couple questions, but I'm going to make just one. So at the beginning of your talk, you set up everything as this spatial setup of the environment can constrain relationships, right? Or what we see as an outcome. And so how do you disentangle this random created relationships based on these spatial constraints for the actual intended relationships between organisms or individuals within a group.
Yeah, I get, so just to make sure I understand, it's the question of how do we disentangle the preference of like the social preferences from the spatial influence? Yeah, that's a great question. I mean, and also I will say that it doesn't have to be constrained. I don't want to be negative here, but it could facilitate, right? So space can also facilitate interactions by providing water, for example, and bringing everyone together. Yeah, I think, Spiegel did a lot of really good work kind of in developing and suggesting different reference models that kind of break those relationships. So I think the way to go about this, there's a few ways to go about this. One is experimentally, right? You can translocate animals and see what happens, right? So this, again, that's why I kind of like, whoa, had a whiplash to my PhD. You translocate elephants to a new place, and they go and preferentially interact with individuals that they knew from before, or that that they have some shared origins with, and with those translocated elephants. And it takes a while for them to integrate into the local society. And so that could be a situation where social preference is more important than any spatial constraints that you throw at them. And so I think, translocations is a very powerful tool to disentangle this and see, is it the rivers that is it just all about rivers and water, or is it about some other social preference? So that kind of disruption. You could disrupt the space they live in. So one of the things I'm hoping to do with the ants is actually put them in different structures and manipulate the spatial constraints on them and see how that influences. So that's kind of the more experimental tools that there probably are others where you can actually break the relationship by changing space somehow, or changing social composition. You've changed social compositions of groups and mix and match. And so I feel like those experimental approaches are extremely powerful to do some of that disentangling. And then in some situations you can, like, you can't always translocate animals. I think in that sense, there's some statistical tools to approach that. And what I mentioned earlier, so Aura developed some really great statistical tools where you can, by creating different reference models that break the different relationships. So if you start breaking kind of relationships in the spatial use, so for example, in his Sleepy Lizard work, he kind of broke the movements over days to see if it's just if the social interactions they see are a function of where animals just move every day, or do they preferentially move to particular places on specific days. And so I think you can also do it statistically by creating reference model where you shuffle certain aspects of the movement patterns or of the social behavior, again, depending on the exact question you're after, and using those informative reference models to compare to your observed to see what changed and how how your observed is different from what were kind of expected or random. Does that make sense?
Yep. Okay, thank you.
Okay, our next question comes from Amanda.
Hello, I'm Amanda Hayesbutt-Farkett, and I'm studying spatial ecology in avian species across Utah right now at Utah State University. And thank you so much, Noah, for a wonderful talk. I really enjoyed it, especially it's merging kind of two of my loves, behavior and species. But I was wondering, have you or has anyone used this tool of the multi-layer networks in using across species relationships? Kind of like, for example, groupings in relation to like threats or other competitors?
Yeah, I think early on in around, I think it's 2014 or 2015, there was a nice review in, I think it's Nature, Ecology, and Evolution by Philosophe is the first author, and Mason Porter is on it as well. Kind of talking about how one might use multilayer networks in ecological system and kind of linking species interactions in multiple layers, where the layers could be, again, different types of habitats, different types of communities, and kind of linking community ecology in a multilayer network structure. So I haven't done that, but I would recommend looking up Shai Filosof's review paper. I think it's around 2014. I can, if you shoot me an email, I can send it to you if you can't find it. Does that, hopefully that will answer your question about how to use it and what to do with it in that sense.
Yeah, yes, thank you very much.
Okay, so our last question comes from YouTube, and this comes from Etsuko Nonaka. He says, they say, I don't know much about ants, but I suppose ant colonies compete for resources and could go extinct. Have you found how social interaction and nest structure relates to competition outcomes?
It's a great question. I have not looked at it, and yes, colonies do potentially compete. They do move around. So the Vermicelliae do move among nests, and so the most closely related stuff that I've done to that question is compared the frequency of nest relocation up north, where I did my postdoc, and down south where I did my where I was a research scientist for a while. And there's some ecological. The reason we compared the relocation frequencies among those two sites was because they're very different ecologically. Well, not very, but they are slightly different ecologically. The southern population lives in a more arid environment. It's much more sparse, so interactions between colonies, there's fewer interactions between colonies. Whereas up north, it's more, there's more water, there's more vegetation, and colonies do interact much more. And it turns out that they move a lot more in the south than in the north. And we think that one of the reasons is basically they're chasing their food resource rather than avoiding either competitors, but also previous work on the population up north, so that colonies will tend to move after they interact with another colony. So it's a combination of kind of, if it's too dense, you can't move enough because you'll bump into others. If it's sparse enough, you can start moving without bumping into others and being able to chase resources and kind of move around where you end up foraging. So it's kind of a combination of both resource use and competition avoidance. So yeah, there's that, but I don't know how, asking whether the actual structure itself. So there are, one thing that we might look at is how, well, and I know some other students in other labs are starting to look at that, is how the nest structure influence alarm. So if an ant is alarmed and starts to spread information about the alarm, how does the spread of that information move inside the nest, and how that's influenced by the nest structure? So one could look at those.
Well, I thank you, Noah. That was really fascinating. And as you can see from all the questions, it was, people were really interested in the work, and we appreciate